In Chapter 1.2, we will discuss about the meristematic tissues and growth. What is meristematic tissues? Meristematic tissues are tissues that have cells division and differentiate to form permanent tissues. There are two types of meristematic tissues, apical meristem and lateral meristem. The first type of meristematic tissues is apical meristem. Apical meristem tissues are located at the shoot tip and root tip in a plant. The cell divides by mitosis to increase the length of plant shoot and roots. The second type of meristematic tissues is lateral meristems. Lateral meristem located at the vascular bundle of the stem and root. Lateral meristems consists of vascular cambium and cork cambium. The cell divides by mitosis to increase the diameter of plant stem and root. In the shoot tips and root tips, there are three zones of cells growth, which are zone of cell division, zone of cell elongation, zone of cell differentiation. The first type of zone of cell growth is in the zone of cell division. Zone of cell division takes place at the apical meristems, which consist of actively dividing meristem cells through mitosis to produce new cells. The second type of zone of cell growth is in the zone of cell elongation. Zone of cell elongation is made up of cells that elongate or increase in size. The increase in size happens through osmosis by water absorption. The water is stored in the vacuoles. The process of small vacuoles fuse to form a large vacuole is called vacuolation. The water absorption exerts pressure against the cell walls, which cause the cells to elongate and enlarge in size. The third type of zone of cell growth is in the zone of cell differentiation. Zone of cell differentiation consists of differentiating cells to reach its maximum size. The cells change their shapes and structures to become specialized cells with specific functions such as xylem and phloem. Phloem and xylem are formed in the shoot tip, while phloem, xylem, and root hair cells are formed in root tip. Now we will look at the types of growth that occur in plants. They are primary growth and secondary growth. The first type of plant growth is primary growth. Primary growth begins after germination and increases in the length of stem and root. This growth takes place in the apical meristems at the shoot tips. At the shoot tips, leaf primordia and shoot primordia will grow to form new leaves and shoots. At the root tips, the root cap will become exhausted when they penetrate the soil. This causes the cells of the root cap to be replaced by the meristem cells. Primary growth starts when the meristem cells in the zone of cell division of apical meristems are actively dividing. The second type of plant growth is secondary growth. Secondary growth increases the circumference or diameter of plant stem and root through lateral meristems. The growth takes place due to the cell division of lateral meristems at the stem and root. Lateral meristems consists of vascular cambium and cork cambium. Next, we will look at the stages of secondary growth occurs at stem, root, and monocots. We will first look at secondary growth at stem. In secondary growth at stem, the vascular cambium is located between the primary phloem and the primary xylem around the pith. The vascular cambium divides laterally to form a cambium ring. The cells in the cambium ring divides to form inner secondary xylem and outer secondary phloem. While the primary xylem is pushed toward the pith and primary phloem is pushed towards the epidermis. As a result, the wall of the secondary xylem are thickened with lignin to provide support for plants. In secondary growth at stem, the cork cambium is located below the epidermis and above the cortex. Cork cambium actively divides to form cork cells on the outer side and cortex on the inner side. The wall of cork cells is waterproof and contains suberin to protect the stem from injuries. Now, we will look at secondary growth at root. In secondary growth at root, the vascular cambium is located between the primary xylem and the primary phloem. Vascular cambium cells divide and combine to form a cambium ring, while the cells in the cambium ring divide to form an inner secondary xylem and outer secondary phloem. The cork cambium under the epidermis divides to form cork cells. Lastly, we will look into the secondary growth at monocots. Even though most monocots do not undergo secondary growth, some do. They are Dracaena species, Aloe species, and Agave species. Here are the importance for primary growth and secondary growth. We will first look into the importance of primary growth. The primary growth causes the shoots to increase in height for photosynthesis. The primary growth also causes the roots to lengthen and anchors the plant firmly to the soil. In primary growth, the primary phloem and primary xylem are formed to help in the transport of organic food, water and minerals to the plants. And now we will look into the importance of secondary growth. The secondary growth increases the stem and roots diameters in a plant. Secondary growth produces secondary xylem and secondary phloem to transport more water, mineral salts, and organic food. The wall of secondary xylem cells is thickened with lignin to strengthen and support the plant. The secondary xylem and secondary phloem also helps to replace old and damaged xylem and phloem tissues. Secondary growth allows plant to live longer and increases the chances of seed production and reproduction. There are similarities and differences between primary and secondary growth in eudicots. Firstly, we will look at the similarities between primary and secondary growth in eudicots. Similarly, both growths involved in increasing the size of the plants. Both growths occur in woody plants. Also, both growths involve cell division in the meristematic tissues. And now we look into the differences between primary and secondary growth. In primary growth, the meristem tissue involved in apical meristem, while in secondary growth, meristem tissue involved in lateral meristem at vascular cambium and cork cambium. As for primary growth, it occurs in both monocots and eudicots plants, whereas in secondary growth, occurs in all eudicots and a few monocots only. The difference in primary growth, it causes the plant to increase in the length of shoots and roots. While in secondary growth, it causes the plant to increase in the thickness of stems and roots. During primary growth, the epidermis, cortex, primary xylem and primary phloem are produced. Whereas during secondary growth, the bark, cork cambium and cork tissue, lenticels and secondary xylem and secondary phloem are produced. Naturally in primary growth, there are no woody tissues in the plant. While in secondary growth, there are woody tissues in the plant. In comparison with primary growth, the bark is thin. In contrast with secondary growth, the bark is thick. Lastly, in primary growth, there is no presence of annual growth rings at the plant stem. While in secondary growth, there are presence of annual growth rings at the plant stem. This is the end of Chapter 1.2, Meristematic Tissues and Growth.